Dana's last name.
Dana, oh my gosh, it's embarrassing. Why I don't remember that right now. Dana, do you have an email from?
No, just I'm getting it.
You would ask me any other time before I was about to do this
Oh, of course,
introduction.
yeah.
Yeah.
Okay. She was going to print my boarding passes for me. Can I send it to you and then you can send it to her?
Yeah, that'll be fine. No problem. A strike. Oh, we got an STO.
Yeah, that I got.
I think we'll go ahead and get started. Hopefully we'll have a few more come in. It's my pleasure today to introduce our seminar speaker, Dr. Mark Kaplan. He comes to us from the Indiana University School of Medicine, where he is the Billie Lou Wood Professor for Pediatrics and also the co-director for the Herman B. Wells Center for Pediatric Research. He received his Ph.D. from Wayne State University in Detroit, and then followed that up with a postdoc at UT Southwestern, and also a fellowship at Harvard in the laboratory of Michael Grusby. And it was here where his career really took off. He made a lot of published a lot of outstanding seminal papers describing a role for STAT transcription factors in T helper cell differentiation. So I joked last night, or referred to him last night as a pioneer at dinner. I'm not sure he enjoyed being called a pioneer, but really is true with some outstanding work. So Dr. Kaplan joined the faculty at Indiana School of Medicine in 1998, where his laboratory has continued to study mechanisms that regulate T helper cell differentiation, including T helper 9 cells, or Th9 cells, which we'll hear more about today. He's published, I think, right around 200 papers, if that's accurate, many in top journals, including Science, Nature Immunology, Immunity, and many others. Dr. Kaplan's received numerous awards, including the Pfizer Schall Award from AAI, also a AAI Junior Faculty Award, and the IUSM Faculty Mentoring Award, which shouldn't be a surprise for those of you who looked at his CV. The list of mentees is pretty impressive. His research has been funded by NIH, including to this day, the American Heart Association, also the Leukemia Society of America. So in short, Dr. Kaplan is a world-class immunologist. We're really fortunate to have him here today. So, Dr. Kaplan.
Thank you very much, and thank you, Ken, for that wonderful introduction. It's really great to be here. I haven't been here before to this part of Virginia, so it's nice to come and experience it. So I mentioned to Ken last night as we were coming back from dinner that I like to incorporate a lot of art into into my presentations. And I think because artists really have a very simple way of looking at a lot of things. And so I know that this is a somewhat diverse audience, and not all of you are immunology experts, and that's okay. Nobody's perfect. But I kind of started off with a really basic concept that T cell differentiation really starts out as Johan Miro diagrammed this years ago, because he understood that the central idea is that a T cell is affected by a cytokine. And that basic aspect of cellular communication is really all that we're going to be discussing today. Now, when I started off in this field, which was a long time ago, this was a model that Alexander Calder put forward, and he understood that there were two types of T cells, Th1 cells and Th2 cells. And for a long time, that's all we really thought there were, from about the mid-'80s until really into the 21st century. But now we know that it's a lot more complicated, and for that we need to go to a model that was generated by one of my favorite artists, Vasily Kandinsky. And he understood from this model that you have a lot of different cell types, and all of these have slightly different flavors. You can see that they all have a lot of similarities. They're all circular, they all have shared some features, but some different among them as well. And really, since this was generated about 100 years ago, all that we've managed to do is add labels onto it. So our accomplishments are really not that exciting. But there's a lot of different flavors of T cells. So from that, we can go to what we think of as being a more standard pathway diagram of T helper cell differentiation. And And as many of you might be familiar, the basic process begins when you have a naive T helper cell that's activated by antigen plus MHC2 on an antigen-presenting cell, and then depending on the inflammatory environment, that naive T cell gets exposed to a lot of different types of cytokines. And those cytokines can drive the differentiation of those cells to various different flavors of cells that we've given these labels to. Those cells then become primed to secrete different panels of cytokines. And those cytokines confer the ability of those cells to participate in different types of immune responses. And this is really the central idea, that one T cell can get programmed to take on these different identities. And this is probably oversimplified too, because there's a lot of interest these days in the idea that there's plasticity of these subsets and they can transit between one and another of the phenotypes. But for our purposes, we're going to take a little bit more simplified approach. So you can see that there is specialization in terms of how these cells provide immunity to various pathogens, and the cooperation in terms of how they generate some of the pathological conditions. And so, for example, whereas Th1 and Th17 may be distinguished in terms of what types of pathogens that they most effectively fight off, like intracellular pathogens for Th1 and extracellular bacteria for Th17, both of these cells can cooperate and contribute to different types of autoimmune disease. In parallel, you can see the Th2 and Th9 both contribute to allergic inflammation and immunity to extracellular parasites, but there are some functions that are distinguished as well, and we'll get into that a little bit in a few moments. So we're going to be focusing predominantly on Th9 cells, and I'm going to talk about a few different stories. In honor of Ken, a lot of it's going to be focused on transcription factors, but there'll be a mix of things, a little bit of stuff that's published, some stuff that's unpublished, some stuff that's hopefully going to get published soon, and some things that are still a little bit unfinished, and give you a little bit of a teaser as to what might come sometime in the future. So to give you a little bit of a background, what do IL-9 and Th9 cells do? And this is something that I think is still relatively unexplored, and we and a lot of other groups are trying to look at this in more detail. But while IL-9 was first cloned back in the mid to late 80s, we still don't understand a lot of what it does. It's been understudied, and I think initially there weren't as many interesting findings of what it was contributing to, and I think more recently some of the functions have been elucidated a little bit better. So as I mentioned on the previous slide, Th9 cells and IL-9 can contribute to asthma and allergic diseases and immunity to helminthic parasites. Th9 cells also seem to play a role in ulcerative colitis, which is one thing that separates them somewhat from Th2 cells. And Th9 cells really provide a lot of anti-tumor immunity. And in a lot of models where they're doing adoptive cell therapies, the Th9 cells seem to be very potent at generating tumor eradication within those models. How does IL-9 do this? Well, that too is really not entirely clear, but in the situation of airway inflammation, you can see that Th9 cells can promote inflammation and accumulation of different cell types, including mast cells and eosinophils, and also can promote other immune cells. And so promotes the survival of innate lymphoid cell, particularly ILC2s. It can promote Th2 responses through mechanisms that we don't really understand yet, and has effects on structural cells as well, which would include leading to mucus production from airway cells, chemokine production from structural cells, changing the effects in barrier function. And so what we've been interested in is some of the transcription factors that are regulating the development of the Th9 subset, and also the regulation of the IL-9 gene. And I know for some of you might look at this and really see something that's more like this, which I think you can see a lot of things in there that are really interesting. But I will focus you on a couple of specific areas that we're going to be talking about. And so the first is is Pu.1, which is a transcription factor that we identified a number of years ago as being important for Th9 development. And so just as a little bit of context for those of you that don't do these experiments a lot, the way that we analyze a lot of these assays is by isolating naive T cells mostly from black 6 mice and stimulating them in vitro under conditions that polarize these cells to the various phenotypes that I've described. And so you have anti-CD3 and CD28 to get polyclonal stimulation and various cytokines that lead to these different flavors of T cells. And then somewhere between day 3 and day 5, we analyze them and do assays like intracellular cytokine staining, chip assays, real-time PCR, and ELISA to look at various parameters. So, Pu.1 is an ETS family transcription factor, and we used mice that specifically had Pu knocked out in the T cell lineage predominantly using the LCK Cree, although there's a few experiments that I'll show you with a CD4 Cree. And they have reduced IL-9 production compared to wild type. And this is some of the data of that. When you look at differentiated Th9 cells over several days of simulation, you can see that there's really a dramatic decrease in the amount of IL-9 that they're producing. And the converse is true as well, that if you overexpress Pu in Th9 cells, it increases IL-9, and if you overexpress it in Th2 cells, it increases IL-9 and decreases Th2. And so it's really one of the best switch factors that we've identified so far. And so we've characterized this in a lot more detail, where Pu is involved in recruiting specific histonoacetyltransferases to the IL-9 promoter. And in various models of area reactivity, we can see that these mice that lack Pu in T cells and so have deficient Th9 differentiation have significantly reduced airway inflammation in a few different models. And this was from the ova-alum model, but we've also looked at house dust mite and others as well. And one of the ways that they promote allergic inflammation is by causing recruitment and expansion of cells, and mast cells are particularly one population that seems to be affected by IL-9. So we wanted to ask a question of whether Pu is really the only ETS factor that's involved in Th9 development. And so I think, as you saw, Pu deficiency results in a pretty dramatic decrease, but not an elimination of IL-9. And so thinking about this, that this theme of transcription factors from similar families working together crosses over into a lot of other areas. And so in immunology, in B cells, Pu and SpI-B, which is another ETS family member, have overlapping but distinct functions. And so the mice that are double deficient have a much more severe phenotype than either single deficient one. And you can see that paradigm in other systems, so in development where other ETS family members actually can cooperate in generating a response or a developmental pathway. And there's a lot of evidence for related transcription factors in T cell cytokine production. So ROR gamma T and ROR alpha in Th17 cells, and TBET and EOMES and CD8 T cells, and how they work in other cells too. And so are there other ETS family members that contribute to IL-9 production? And the way we got into the first candidate that we looked at was a little bit backwards, but I'll lead you through it. And so the ETS family is relatively large. There's almost over 20 proteins that fall into this family, and they all have the ETS DNA binding domain. And then there are subfamilies that are based on structural similarities and also DNA binding specificities. And so Pu is up here at the top, and this one that I'm going to be talking about first is ETV5, and ETV5 is we identified it because it was known to be induced by STAT proteins, and this was work that was done back in the 1990s by Ken Murphy's lab. where they showed that it was a STAT4 inducible factor, and they suggested it was having some effects on Th1 cells. We got interested in this because it was interacting with another factor that we were interested in, and that work developed to show that ETV5 has a really important role in terms of IL-17 production in Th17 cells. And what we showed is that it had no effect in Th1 cells. And in Th17 cells, it was very restricted, that it affected cytokine gene expression, but not the expression of a lot of other of the Th17 associated genes. And so what we noticed when we looked at a house dust mite model in those mice was that they had reduced IL-9 production. And so given that Pu deficiency reduces but doesn't eliminate Th9-mediated IL-9 production, we hypothesized that ETV5 functions in parallel with Pu to regulate differentiation. So we tested that first using, in this assay, intracellular cytokine staining, where we differentiated Th9 and Th2 cells, and you can see IL-4 on the Y and IL-9 on the X-axis. And so wild type, you can see that there's almost 40% that are IL-9 positive, and in the ETV5 knockout CD4 cells, it's reduced by about half. And you can see the much lower amount of IL-9 that you see in Th2 cultures is also decreased in the absence of ETV5. You can do the converse experiment where you transduce ETV5 into cells, and so when it's overexpressed, you see that there's about a twofold increase in IL-9 in the Th9 cells, and also about that in the Th2 cells, although much lower because they're not really primed. So to test the question of cooperativity, we crossed those mice together so we could generate Pu and ETV5 double knockouts. And the results there were pretty rewarding. And essentially, so here again is kind of the data that there's about a 50% decrease in the ETV5 single knockout. There's a little bit more than 50% lost in the Pu knockout. And when you have both of these factors gone, then you really see that there's a dramatic decrease in the amount of IL-9. And this can be averaged over several experiments, and you can see that that the double knockouts really have a decrease in the amount of IL-9. So from there, we wanted to look in vivo. And for this, we did the ova alum model, where the mice are immunized twice with ova alum and then challenged intranasally over a week or so. And this generates a fairly strong inflammatory response in the lung. And so what you can see here is these darker pink areas are areas of inflammatory foci. And you can see that in the Pu knockout, a lot of that is lost. Not so much in the ETV5 knockout, which is somewhat interesting. In the double knockout, it's hard to distinguish it from the Pu single knockout. When we looked at mucus production, we saw something a little bit different. And so this is PAS staining. The wild type airways, you can see that there's a lot of mucus production from the goblet cells. In the ETV5 knockout, this is almost completely lost. In the Pu knockout, partially lost. And the double knockout, you really don't see any, as you would expect, so kind of accumulation there. So what Pu seems to have greater effects on infiltration and ETV5 on mucus production. And you can plot this out looking at specific things in vivo. And so in vivo, you can also see this stepwise decrease in the amount of IL-9 that's being produced within each of these mice. And so in the double knockouts, having the least amount. But if you look at eosinophil recruitment, you really see that ETV5 doesn't have much of an effect, Pu decreases it, and the double knockout really not that much different than that single. Whereas if you look at mast cells, each of the knockouts seems to have some effect on the amount of mast cell accumulation within the tissues. So similar to Pu, ETV5 expression promotes Th2 differentiation. And data that I didn't show you is that ETV5 expression seems to be regulated by both the IL-4 and the TGF-beta signals in generating Th9 cells. Co-deficiency of both factors has an additive effect on IL-9 production, both in vitro and in vivo. But what we saw was that there seemed to be some separation of their function, which likely means that they're controlling separate things within the Th9 cells that ultimately lead to different phenotypes when you're looking at the allergic inflammation models. So we'll go to another artist example and thinking about lineage-defining transcription factors. And we know a lot of these from work that's been done over the years by many groups, that TBET, for example, is associated with Th1, GATA3, Th2, et cetera. And what we still don't really understand is what is the factor that's most lineage defining for Th9 cells. And the data that I've described for Pew is probably the best so far in terms of having an effect that is specific within the Th9 lineage. There's other factors that we and others have published on that are important for IL-9 expression, but they're not really specific for the Th9 lineage. They can affect a lot of the other lineages as well. And so we wanted to try and get at this question. And the one thing that we know about Pew is that while it is important, it is not very differentially expressed. And so if you look in Th9 cells, sometimes you can see maybe a twofold increase compared to some of the other populations. But it's not as differentially expressed as some of these others like TBET or GATA3, where you can see a 5 to 10-fold enrichment within that subset compared to some of the other subsets. So to get at that and some other related questions, we did some microarray analyses, and this was done a number of years ago now. And we did the comparisons with Th2, Th9, and T regulatory cells. And the reason we chose those three, to simplify it a little bit, was because, as I've noted, Th9 cells are differentiated with IL-4 and TGF-beta. And Th2 cells differentiated with only IL-4, Tregs with only TGF-beta. And so we're looking at the two cells that have single cytokines and then what happens when you have them together. And I think this also gets to a question that was in the field of whether the Th9 cells are really an individual subset or whether they're just a subset of the subset of Th2 cells or another differentiation state of Th2 cells. So you can, just by looking at the colors, you can see that Th9 and Th2 are far more related to each other than either of them to T regulatory cells. But the important thing is, when you really hash this out, is that there's about 600 genes that are at least twofold upregulated in Th9 cells than either of the other subsets. And just over 200 that are at least fivefold enriched within Th9 cells compared to Th2 or T regulatory cells. And so we looked at a number of those, and these are some of the ones that were most differentially expressed. And so this is IL-25 receptor, this is the IL-1 receptor antagonist, and ERG was one of the other proteins that caught our eye. And that comes back to the ETS family. And so ERG is another ETS family protein. It has some unique domains, and it's even more complicated than that. There's at least 5 different ERG isoforms in humans and 8 in mice, and these are all from splicing isoforms, so they're all from the same gene. ERG3 is what's commonly known as ERG, but it's a little bit hard to actually piece apart what these different splice forms are. And what makes this a little bit more difficult to study is that that homozygous deletion of an exon encoding the 5 to 8 isoforms is embryonic lethal. And so that means that there's going to be some additional approaches that are needed to really looking at function in vivo. So we wanted to do some initial experiments to see if this really had a function in Th9 cells. And so this is flow cytometry doing intracellular staining for ERG. And so you can see isotype staining here in the control retrovirus in the shaded peak, and if you overexpress it, you can increase expression of ERG. And what happens when you overexpress it is you get an increase in IL-9, and that's shown here where the amount of IL-9 in the cell population is almost doubled. And you can also see that there's a big increase in the mean fluorescence intensity of the cytokine staining. And so that means that it's not only more cells that are making IL-9, but more IL-9 per cell that's being made. And so then we did the opposite experiment, where we knocked down ERG. And you can see that again here with the control RNA, siRNA, and the ERG specific one, and that decreases expression. And you can see that the amount of IL-9 being produced by these cells is diminished when you knock that down. And this is just averaging out several experiments, looking at the percentage of IL-9 positive cells and the MFI, which is significantly but very modestly decreased. So we then come to the question of how do we piece together how these three factors might actually be contributing. As I'll say in a minute, we still don't have all of the answers because we're still trying to generate an ERG deficient mouse that will allow us to answer some of these questions. But we did do this experiment where we overexpressed each of these factors either individually or together. And so you can see that ERG, PU, and ETV5 pretty similarly stimulate IL-9 production from those cells, and this is just averaging fold over control of the amount of IL-9 that they're producing. And I will say that we have identified some ETS family factors that don't have this effect. So there is some specificity. It's not an artifact per se. But what we did see is that when you put ERG and PU together, you really have a significant increase in the amount of IL-9 that's being produced. And a little bit of an effect with ERG and ETV5, but not as much, not significantly. And PU and ETV5 also really not an effect. And if you put all three of them together, it's no different than putting ERG and PU together. So it suggests that those two factors are somewhat unique in their ability to be able to mediate induction of IL-9. And so that's something that we're...
Just ask a quick experimental question. So is the overall level of Xfactor roughly the same between when you overexpress all three versus one factor?
And I'm not sure if I have the answer to that.
Okay.
So certainly, I think ERG was. ETV5, we haven't been able to intracellular stain for, so it's not entirely clear.
I have a question.
Sure.
So, based on your formula showed that double knockout ETV5 and the PU.1, when you double knockout compare the single knockout of PU1, the bigger difference not that big, but when you co-treated with ERG with PU1, but you claimed before these two are parallel. So then when you double treated with ERG1 and PU1, looks like the more significant is the ERG1 is more important than ETV5, because if you compare your
Sure.
tripled with the double the ERG with the PU1, it's pretty similar.
Right. Right. So I mean, I think I would agree that what we're proposing and what we still don't have the final story for is that ERG is more important than ETV5 and possibly more important than PU.1. But I think as we generate those conditional mice and as we actually cross them into some of those to make double knockouts and single knockouts, we'll be able to figure that out for sure. So basically, just to sum up a few things, so ERG is preferentially expressed in Th9 cells, and that's something that a lot of the other factors that regulate IL-9 are not, don't have that feature. So STAT5, STAT6, SMADs, IRF4, BITF, ETV5, they're not really differentially expressed among the subsets. As overexpression increases IL-9, and shRNA for ERG decreases IL-9 within those populations, and there is evidence of some cooperation with PU.1. So as I alluded to, that we're now in the process of developing some ERG conditional mutant mice that should allow us to ask some of these questions. And at least what we would propose, coming back to this slide, is that maybe ERG is really the critical factor that's important here. Okay, so from this story, we're going to move on to a slightly different one. We'll come back to this slide, and so rather than thinking about the transcription factors that are leading down to this, that thinking about the IL-9 gene and focusing in on regulatory elements within the IL-9 gene. And so a lot of this is really just cartooned around the promoter and thinking about the direct activation of IL-9. We've been interested in trying to think about what other regulatory elements might be there. And so one of the ways of trying to do this is to look at conserved non-coding sequences, or CNS. And these are regions where if you compare sequences and genes between different species, you can see that there's conservation of those sequences where you don't have any actual coding sequence, and that would suggest that there is some conserved function there. So we identified a few of these that are relatively close to the gene. This is the gene up here, the motor that's conserved to be a prime that we called C, and from the promoter. Didn't seem they seem or don't. And this binding